And we're going to pick up now with our second section of our discussion of Chapter 30 in the Raven textbook on the green plants. So, moving on now into the bryophytes, which are the first and earliest and most primitive group of plants. These are non-tracheophyte plants because they do not have true vascular tissue. They do not have tracheids, hence the non-tracheophyte plant. They do have some rudimentary very relatively inefficient water conducting tissue, but they certainly do not have tracheids. They are also the first group of plants to develop what are called mycorrhizal associations. And if you think back to Principles 1, mycorrhizae, this is a type of symbiotic and mutualistic relationship whereby a fungus enters into a relationship with the roots of a plant, and both parties begin to benefit each other. So the mycorrhizae are going to get carbohydrates from the plant. The plant is going to get phosphorus from the fungus. It's going to get increased water uptake from the fungus. Both of those two functions are due to the increased surface area that the fungus provides either in or on the roots of the plant. And the plant is also going to get some protection against other nastier, more pathogenic types of fungi which might otherwise harm the plant. So we see mycorrhizal associations in roughly 90 to 95% of all land plants, and really it's a must, it's obligate. If the plants don't have the mycorrhizal fungus, they're not going to grow very well at all, if they even can survive. It's a pretty tough existence without those little fungal partners. The bryophytes, relatively big group, nowhere near what the angiosperms are going to be, but it's about 25,000 species. And they can live in a relatively decent array of different habitats. This picture here is of a hornwort, one of the ones that's probably the least familiar of the three bryophyte types to us. But these three groups, liverworts, mosses, and hornworts, can live in some fairly variable kinds of environments. One thing I want to point out too, the term bryophyte can be used in a couple of ways. Bryophytes can be used as kind of a common name, almost like a group name. Like mammals, for example. Bryophytes can be used in that context as well, in which case you're talking about really any of the seedless non-vascular plants, liverworts, mosses, and hornworts. However, Bryophyta, with an A at the end of it, is the name of a phylum. The Bryophyta are the mosses. And in that context, it actually is a real taxonomic name of a group, in this case a phylum of plants. So in this case, bryophyte and bryophyta can be used to describe both sort of a more casual common name for a big group of plants, or more specifically, if we say bryophyta, we're referring to a phylum of plants that include just the true mosses. So as we said before in the previous section, in the bryophytes, the gametophyte is by far the dominant generation. It is going to be green, it is going to be photosynthetic, it's going to be around almost the entire portion of the plant's life cycle, whereas the sporophyte is going to be much smaller, much more temporary, and in most cases completely dependent on the gametophyte for its entire existence. In this plant here, in this hornwort, these little spikes up here, or horns as they're sometimes called, are the sporophytes. Hornworts actually do have a photosynthetic sporophyte, whereas the mosses and really the liverworts don't. Their sporophytes are more or less non-photosynthetic, but the hornworts have a photosynthetic sporophyte. The gametophyte is the darker green kind of part growing down around here. This part is photosynthetic, and it's around all the time. It's actually doing the bulk of the support for this plant's life is happening down here in the gametophyte. So the haploid generation is definitely the dominant one once you start looking at bryophytes. As I said before in the previous section again, the bryophytes, again referring to the entire group of 25,000 species, is kind of, in a very loose sense, the plant equivalent of what amphibians are. They were able to move out of the water to a degree, but they still have to be near water or have water around them at the very least for the sexual reproduction part of their life cycle. They have to have water for reproduction. So we'll take a look at this phylum first, the phylum Hepaticophyta. Hepaticophyta are the liverworts. So now we're talking about, we're in the kingdom Plantae, or kingdom Viridiplantae if you want to go with a more recent classification. Kingdom Plantae, phylum Hepaticophyta. Now if you look at this prefix up here, hepata, okay, this refers to liver. Like hepatitis, for example, refers to a disease of the liver. These are liverworts. The reason why they're called that is because the gametophytes, which are kind of shown down here in the bottom part of this picture, this and this part up here and this, that is the gametophyte. And when you see the entire plant, which you don't really in this picture, so it's a little bit awkward, they look kind of sort of like lobes in a liver, even like in your fetal pig livers that you guys saw. They look vaguely liver-like. And so back in the old days of medicine, the bad old days of medicine, some of these liverworts, because they looked like a liver, were used medicinally. They would be used with the idea in mind that if you have a part of your body that is ailing you or hurting you, or if you're sick for some reason, if you could go out in nature and find something that looks like that part of your body and consume it, it's going to make you feel better. Now, clearly this didn't work in the vast majority of cases, but this was kind of what people went with. This was the state of medical knowledge back in the Dark Ages. So this is what people did. Now, that being said, a lot of liverworts don't have that liver-like appearance, but the ones that we talk about kind of most frequently, at least in General Biology 2, Principles of Biology 2, is the classic Marchantia as one genus that looks a lot like the classic kind of liverwort. The gametangia in this case, the parts that are actually producing the gametes, remember gametophyte looks, it's haploid, it's going to contain the gametangia, which are eventually going to produce the gametes. They look kind of like little umbrellas in this case. These little structures up here that look really a lot like a little miniature palm tree are producing the female gametes. They're producing eggs. The male gametophytes, gametangia, are going to look a little bit different. They're going to look less like a palm tree, more like a disc or like a frisbee at the top of a stalk. They're going to be producing sperm cells, but it's kind of the same idea. This is the gamete producing part of the gametophyte plant. And so if you have water around and you give those little sperm cells a chance to swim to the egg cells, you can actually have fertilization. And so in this case, you can't actually see it in this picture, but if fertilization were to occur, there would be a sporophyte that would form up underneath the little like micro fronds of this little microscopic looking palm tree. There would be a sporophyte tucked up underneath there. Very, very temporary, very, very short-lived, but it would be diploid and it would be multicellular, producing spores which could then be dispersed out and available then again to form another gametophyte plant. In the background of this picture, you can see these little cups back here, and these are called Gemmy cups. Gemmy is spelled Gemmae. Gemmy is Gemmae. If you're playing along at home, Gemmy cups are for asexual reproduction. And so what these are for is if it is raining, and again these things often live in humid, rainy kinds of environments. If it's raining, if raindrops come down and splash the inside of these little cups, there are little clonal plants that can be splashed out of the cup into the surrounding soil where they can establish and begin to grow again. And so you can actually find a relatively decently sized patch of liverworts that genetically are all the same because they formed from one initial parent plant. The second phylum within the bryophytes is the Bryophyta itself. This is the biggest of the three phyla. In this case, we have what traditionally you would consider to be a moss. Maybe mosses like you have growing even in your own backyard. The gametophytes are never really big. Again, they don't have water-conducting tissue, so they can't ever really get very big. But they might stand a couple, 3-4 inches tall, maybe at most. They have a single, what looks more or less like a stem. They have little, what look like leaves radiating out away from the stem. I should say they're stem-like and leaf-like because they do not have true vascular tissue. And again, to be considered a root or a stem or a leaf, it has to have actual vascular tissue, which these don't. The roots in this case aren't roots. They're what are called rhizoids. They're root-like structures. They really don't do anything to absorb water at all, but they do serve an anchoring function, much like a root would do as well. So the rhizoids are going to anchor the plant to the substrate where they can attach and stay for a relatively long period of time. The gametangia in this case are going to be a bit different. And actually in lab, you should have drawn or started to draw these, trying to think about where we are at this point. A male and female gametangium of a neem plant, or a nimium plant. So we have Archegonia and we have Antheridia. The Archegonia are always those that are producing the female gametes. In Archegonium, is a gametangium that produces female gametes. I guess you could reverse that, but I'll say it again anyway. An archegonium is a gametangium, a gamete-producing structure, that produces female gametes, which would be eggs, right? So we have an archegonium up here. If you look all the way up here at the tip of this plant, you find these little archegonia. And if you zoom in even more, you see these little tiny egg cells tucked up inside those archegonia. And you will have drawn that, or you're going to draw that if you haven't already. If we look at a male plants tend to have more of a flat top across the top, whereas females are more of a spiky top. If we look all the way at the top of this male plant and zoom in, we see antheridium. Now, an antheridium is a gametangium that produces sperm cells, male gametes. So we see these antheridia, which produce sperm cells, which can then be used to fertilize the egg cells, in which case we get a zygote. So now we have not an egg cell, but a zygote. This is now a diploid cell inside the archegonium, which is going to begin to grow upwards and outwards. And eventually, in a lot of mosses, what we're going to see are these capsules growing out of the top of the female plant. This female plant here, starting off with a zygote, produced an embryo, which grew upwards and outwards until eventually we see a stalk with a capsule at the end of it, and inside this capsule we're eventually going to find spores. Okay, so this entire stalk and capsule is diploid tissue because it started off from this embryo, which is diploid. The embryo started off as a zygote, which was also diploid. So we know this entire thing has to be diploid, 2N. And eventually this diploid structure is going to start having cells that undergo meiosis. And like any other time when we have meiosis, what that means is that these diploid cells inside the sporangium are going to become haploid. The sporangium can then open and disperse these spores out in the environment where they can start to undergo mitosis, and eventually give rise back either to a female gametophyte or a male gametophyte plant. Okay, there's a lot there, but I definitely want to make sure you pay attention to this life cycle. You've seen this in lab, you have drawn this, we've talked about it quite a bit already in this lecture, so you probably should have some indication that I might think this is kind of an important point. Again, these plants must have water around because the sperm are flagellated, so they need to be able to swim to get to where they need to go in order to fertilize an egg. Right, so here's just a bigger view of that life cycle. Male and female plants with antheridia and archegonia respectively, sperm cells and egg cells respectively. Fertilization occurring, we've got a zygote, N plus N is 2N. Zygote gives rise to embryo. Embryo eventually produces a stalk and a capsule. Cells inside the capsule / sporangium, same thing. Capsule, sporangium are the same thing. Cells inside of here go through meiosis. The capsule opens up. Spores are released. Some of these spores are going to become male. Some are going to become, these are going to become male. These are going to become female, eventually giving rise to gametophytes, which are female or male, respectively. The last group of bryophytes that we're going to look at are the Anthoceratophyta. It's kind of a mouthful, Anthoceratophyta, which are the hornworts. This is a group which seemingly kind of sort of comes out of nowhere. Now, both the true mosses, the Bryophytans and the Hepaticotophytans, the liverworts and mosses respectively, let's go with that because it's a lot easier to say, they've been around for much, much longer. But then all of a sudden, during the Cretaceous, which is around the same time where dinosaurs were kind of big and doing their thing, the end of the Cretaceous is when most of them went away. Right? We talked about that. All of a sudden, hornworts show up. And we don't really know where or how they arose. This is the group, like I showed you back in that first picture, that has the photosynthetic sporophyte. So down here, this kind of almost looks like jello in this picture. This dark green stuff down here, this is gametophyte tissue. This is producing gametes, male and female. And then these stalks, or if you want to call them horns, are growing out of that. These are the sporophytes, which are diploid and then available to eventually go through meiosis to produce more spores. The sporophyte, again, like in the mosses and also in the hornworts, is completely dependent on the gametophyte for all of its nutrition, its support. Although in the hornworts, it does get its own carbohydrate because the horn or the sporophyte itself is somewhat photosynthetic. Couple of bizarre things about this group. Number one, the cells in these actually have one chloroplast, which is kind of weird. If you look at an angiosperm plant, like a maple tree, right, the leaves are going to have many cells. Each one of those cells might have anywhere between 40 and a couple of hundred chloroplasts. In hornworts, they don't. The cells only have one big chloroplast, which is kind of unusual among the plants to have that kind of a structure. Let's see. Okay, so one other kind of unique thing about the hornworts is that the sporophytes actually grow differently from any other kind of plant tissue. Right? Back in the last chapter, in 36, we talked about primary growth, and we talked about how plants grow. They only grow at the tips, and they only add new cells at the tips. The cells that are further down toward the base, they actually don't change position. They stay where they are. They don't go anywhere. They don't move. But with these, with the sporophytes in the hornworts, the new growth happens down here at the base. And when new cells are added down here, that's going to push the upper parts of the sporophyte away from the gametophyte. So it's actually growing from the base as opposed to growing from the tip, which is the custom and kind of in most plants. All right, so we're going to move into the tracheophytes. These, again, tracheophytes are plants that have tracheids. They have true vascular tissue, and they begin to show up about 420 million years ago, kind of around the same time where animals had already kind of diversified, and we've already seen a lot of the main animal groups by this point, but they're all there at least. It's really kind of an important time in the Earth's history. The Rhiniophyta is a group which we think, we're almost sure, were tracheid plants, or they had tracheophytes. They were relatively small. They apparently didn't have roots. They apparently didn't have leaves. Relatively simple, relatively rudimentary, but based on fossils that have been found, it does appear that this group did have the ability to have vascular tissue. They were also homosporous, which means that the spores they produce were all homogeneous. In other words, if you had a male spore-producing plant or a female spore-producing plant, they would look identical. You couldn't tell them apart. With most of the modern-day tracheophyte plants, they do look different. The male and female spores are different enough. You can tell what they're going to become. Think back before we talked about megaspores and microspores. Megaspore mother cells, microspore mother cells. They're very different. One's a lot bigger than the other one. Not so in the case with these early tracheophyte plants. They actually are homosporous, which is kind of similar to what we're going to see in the ferns, for example. All right, so just to kind of end here quickly on this section before we get into the tracheophytes, xylem, remember, is water-conducting tissue, starting off with tracheophytes in the earlier, or starting off with tracheids in the earlier tracheophyte plants, and later on moving into vessels. These are transporting water. Phloem cells are going to start off as sieve tubes and sieve tube members. And both of these two tissue types allows us to get plants that are just a lot bigger, larger size, a lot taller, a lot bigger in diameter. Some of the bigger sequoia trees and coast redwood trees. The sequoia tree is like in this picture here. These can be quite large in diameter around the base. Coast redwood trees can get upwards of 350, 360, maybe even upwards of 370 feet tall. That's quite a bit longer than a football field if you're keeping track at home. That's a very large tree. They couldn't do that unless they had vascular tissue. For two reasons. The vascular tissue gives them strength. We talked about secondary xylem before in chapter 36, right? So we get strength from secondary xylem, and we also get water conduction from secondary xylem. And without those two things, plants would have stayed really small, like most mosses are. Again, this is going to be the sporophyte. The gametophytes typically don't ever really have much in the way of vascular tissue. We also started seeing things like a cuticle and stomata when we start moving into the tracheophyte plants, even into the ferns and some of their relatives or their allies. The cuticle is definitely there. The stomata is also there in a lot of those other early tracheophyte plants. All right, so we're going to stop this little section of lecture here.